lung cancer is the leading cause of cancer-related deaths, and really it's quite rare that a patient would get that diagnosis and then have a treatment that would be effective, and therefore this is really why we've focused on that in terms of the type of cancer we've been investigating. Another really important thing about lung cancer in the context of our research is that it has a really prominent tumour microenvironment, so it really changes the cells around the cancer, and this has a really profound effect on how aggressive the cancer might be. So it made it a really relevant cancer type to look at when looking at the tumour microenvironment. And so what we've known for a number of years is that these cells called fibroblasts can be hijacked by the cancer in order to help it to grow. But what we haven't really known is the different types of fibroblasts that you can find and the different ways that that process can occur. And so what this research has been able to do is identify these different types of fibroblasts that you find in the lung and how they change when they come and interact with the cancer. And so what this has really shown us is that you get these three different types, and one particular type called myofibroblasts are the ones which are seen to be associated with patients who do particularly poorly. So their survival rates are a lot shorter than those without the myofibroblasts. And conversely, the other two types of fibroblasts that we found actually go the other way, and they are correlated with better survival for patients and potentially therefore are doing something to prevent the cancer from growing. And so this kind of dichotomy in how fibroblasts might affect tumour progression is something that we haven't really been able to either investigate or understand to any great detail before. We've studied these cells, fibroblasts, for a long time now, and what was quite striking about them was the fact that they seemed to do lots of different things in the tumour. And what we couldn't necessarily understand is how one cell could do so many different things, how it could carry out so many different functions. And so that kind of made us think perhaps there are different types of fibroblasts that you find in these cancers, but it was very difficult to be able to study that, and it was particularly difficult to study that in human tumours. And so what this new technology has enabled us to do is take samples from patients where they've had their tumour removed due to, like, from a surgical resection, and then analyse it in this really unparalleled detail. And basically that comes about from having interdisciplinary teams involving, we as cell biologists need the help of engineers in order to build the equipment that enables us to take apart these tumours and isolate these individual cells and extract this crucial information that we are analysing about them in order to learn about their different functions and what they do within the tumour, and ultimately, hopefully, how we can target them. This type of fibroblast, this myofibroblast that we found in our study, is something that's not necessarily specific to lung cancer, but also will be found in different cancer types. And so we actually have a new grant from CRUK where we'll be analysing these cells in more detail in head and neck cancer and trying to see what they do there as well as lung cancer. And we hope and expect to find some quite conserved functions across these different cancer types. And if we look to targeting these cells therapeutically, then of course that extends the range that could be effective for.